this evening. I know everyone has very busy schedules and taking time out to listen to a presentation. That's very, very kind of you. So hopefully, as Tracy said, this is about providing information for you to then decide whether you wish to take a vaccine or not take the vaccine, or give you the confidence to be a bit more informed about the choices that you have. So I'm just going to do a presentation. Unfortunately, I couldn't do it as shorter than 15 to 20 minutes, but I will try to do it as quickly as possible with enough details that you can gain some information and knowledge about how vaccines are produced and the new vaccines that are rolling out, why there's so much talk about them, and any of the concerns and worries that you have. We can answer in the questions and answer session at the end. So I'm just going to start the presentation. And hopefully you'll be able to all see hopefully you can all see the presentation now.
Yeah, that's great. Thanks, Raj.
Great. Okay. As I said, this presentation is not an official NHS or government presentation. This is just an opportunity for us to give you some information about how vaccines work and dispel some of the myths and news out there, and to give you some explanation of how the immune system works. Okay, and I have tried to simplify as much as possible, but please bear in mind this is a really, really complex field, and technically it is still, a lot of research been going in this, but it's evolving and changing as we go along. So it may not be 100% accurate, but it's a good summary, I hope. So the presentation today will cover 5 main areas. First area we'll cover is about the natural immune response, and then what the traditional vaccines are that we've been using for many years, and the new generation of biotech vaccines that are coming out, and discuss some of the pros and cons. And then hopefully we'll try to bust some myths around some of the vaccine rumors and fake news out there in the media. In this video, I just want to do a very short, in this presentation, I just want to do a short video, and this was produced by the NHS's National Institute of Health Research, which tries to explain in a summary about what is the immune, how do vaccines work, and to give you a brief kind of background information. So if you just hopefully listen to the video for a few minutes, and then I'll come back with the presentation afterwards.
To understand how vaccines work, it helps to look first at how the immune system works, because vaccines harness the natural activity of your immune system. There are about 100 trillion bacteria and viruses on your body. Not all of them cause disease, but some are able to get inside our bodies to multiply, and this can make us ill. There are barriers to stop this happening. But suppose some disease-causing bacteria do get through. Your immune system is quick to recognize them as invaders. This is because the proteins or sugars on the bacteria's surface have different shapes to any of the ones in a human body. They trigger a complex chain of events involving many different types of white blood cells working together. One type of white blood cell is able to make antibodies to fight the invaders. Antibodies can stick to the proteins or sugars on the bacteria's surface, and this kills the bacteria or disables them. However, not all antibodies will work against these bacteria. They have to be exactly the right shape, a bit like a key fitting a lock. Our bodies have a library of billions of white blood cells, each of which can make just one shape of antibody. Only a few of these antibodies will match the invading bacteria. Producing antibodies of the right shape can take several days. By this time, there could be billions of disease-causing bacteria in your body. Once the right cells are activated, they quickly divide and turn into a production line, making masses of antibodies that stick to the bacteria. Eventually, your body gets rid of all the bacteria and you recover. Antibodies remain in the blood, and some white blood cells may also become memory cells. If those specific bacteria invade the body again, the immune system will respond so quickly that you won't get ill. Vaccines work in the same way. They contain weakened or dead bacteria or viruses, or even just a few proteins or sugars from the surface. This is enough to convince the immune system that a real invader has got in. The same process takes place as when real bacteria or viruses invade our bodies, except you don't get ill. Afterwards, if your body ever meets the real thing, your immune system will remember it and get rid of it before you even know it's there.
I think that was hopefully a really nice summary of how the immune system works, and it kind of encapsulates a lot what the following slides are going to go in a little bit more detail. But it's just to remind ourselves that even though the video mentions a lot about bacteria, this also applies to viruses. So if we come across virus that we haven't seen before, our body will respond usually in the same manner as described in the video just gone. But there are occasions when we haven't seen that virus before, or it's a novel new virus, and our body may not be ready to respond in such a strong response straight away. And that's where vaccines come into. And the next slide, whoops, I just wanted us to quickly remind some of us and all of us, and especially some of the younger people who may have not had an experience or memory of some of the devastation that viral infections can cause. And not only the impact on people, but on society as well. So one example is smallpox, which in the 15th to the 18th century was one of the most deadliest human diseases known and killed 3 out of 10 people. And millions across the world were killed and died from smallpox following the expansion of European settlement across the world. But in 1796, an English doctor named Edward Jenner recognized that actually he found that milkmaids who had been exposed to a similar related virus called cowpox, which didn't cause it only caused a mild form of the disease. If you had those milkmaids who had cowpox, they never caught smallpox. So he then discovered that if you took part of the cowpox and rubbed it into a small wound on a person who didn't have cowpox or smallpox, then they would not get the deadly smallpox. So this is the beginning of the notion of vaccination. We could find that if we exposed our body to a related or similar organism or bacteria or virus, our body can generate an immune response to protect against a much more deadlier form of the virus. And another example as shown in this slide here is about polio. Again, in the early century, polio went on to devastate many, many lives. Over 650,000 people could have been killed or probably were killed over the years. And also before the polio vaccine came along, polio was the major cause of disability up to about 1960s. And in the 1960s, when an American doctor, Salk, developed the first polio vaccine, we started to free ourselves from this horrible disease. And along came Dr. Sabin in 1961 with a much more easier to use oral polio vaccine, which I think probably people remember having maybe in their childhood. And this, as you can see from this graph, has resulted in, from 1988, where polio was still endemic in many, many countries around the world, to in 2015, there's only a few countries where polio remains an endemic disease. And hopefully within a few years, or probably now, very, very soon, polio again will be eradicated by vaccination and no longer be a disease that cripples and kills people throughout the world. Now, there are lots of different types of vaccines, and I wanted to try to group them into at least 3 categories. The first type of vaccine, and all vaccines, as explained in the earlier video, is designed to mimic the immune response, mimic and trigger an immune response in the body. And one of the best ways to do it is to use a live virus. So these are also called live or attenuated vaccines. So they use the live virus or a version of the virus which has been weakened, so it doesn't cause some of the deadly part of the disease that it's meant to cure. So these live vaccines remain some of our best and strongest and longest lasting vaccines. However, they're not suitable for everyone. If you have a weakened immune system or some underlying health conditions, the fact that they are still contain live virus in there can cause a flare-up of the disease, and you could end up getting the disease rather than the immunity. So even though these vaccines are very good, scientists have tried and continue to struggle to find new ways and better ways of trying to produce a vaccine which is both more safer and more effective. And we then go on to the next kind of category of vaccine, which are called, which are either whole viruses which have been killed, or where the vaccine uses parts of the virus. So small parts of the virus, the protein, the sugars on top of the viral surface are used to trigger the immune response. And these vaccines, because they've got no live virus in them, or they've only got parts of the surface protein of the virus in them, they are much, much safer. There's no chance, or very, 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 very small chance of catching any disease. So the safety of that vaccine is much, much higher. However, the immune response is weaker. And we usually need to require repeated booster doses. And these can be months to years apart. And they are quite hard and expensive to produce, because you have to grow lots of the virus in other cells, in another host, in another animal, and then purify it, and then kill it, and then inject it into the as a vaccine. And I'll tell you why we think, and later on, and I'll explain in some of the slides, why the immune response to these types of vaccines is generally much weaker as we go through the talk. Then we come on to our third type of vaccine, hopefully, which is the newer and more advanced vaccines called the recombinant or vector vaccines. These now we're starting to get into much more technical, complicated science in trying to improve the vaccines. So in these types of vaccines, the DNA, which is shown here, which encodes the genes for some of the virus. So it's not the actual virus, it's not the protein of the virus, it's the gene that encodes that protein, is put into a host cell, which then makes more copies of the protein that we're going to use to raise antibodies or create the immune system response. This purified protein is then injected as the vaccine. The advantages of this is you can use host cells such as bacteria, yeast cells, and nowadays we can grow animal cells in the lab, and they can be used to produce lots and lots of this protein in large quantities. But again, what we find with these vaccines is that the immune response is weaker, and again, you need repeated doses to maintain immunity over a long period of time, and they do take longer to develop because you have to know all the sequence of what gene that you're going to use, and the technology behind it is very complex to scale up. So this brings, I'm just going to go in a little bit more detail about recombinant vaccines. And when we do that, we need to think a little bit about how the cell shown here produces proteins, because when we produce in a vaccine, we usually produce in a vaccine to a protein that is part of the virus. So normally in a cell, you'll have the nucleus, which contains your DNA. That's deoxynucleic acid, ribonucleic acid, and that nucleus, which contains the DNA, then produces what's called a messenger RNA. And this is like a messenger that goes into the cell, which then directs the cell to produce the proteins that are used to drive the function of the cell. Now, the proteins are all produced depending on the different functions of the cell. So your heart cell will want proteins that help it to beat. Your lung cells will have proteins and specialized machinery to help it absorb oxygen. And this is all dictated by the genes and the messenger RNA that's produced by the cell. And one key thing I think that we really need to remember for later on in this talk, because we're going to be covering RNA vaccines, the key factor to remember in these is that even though DNA, deoxyribonucleic acid, and ribonucleic acid, the mRNA, they sound very, very similar, and they may look similar. They are completely different molecules. They are as different as apples and donuts. They're not even close to oranges. So we know that in the cell, in the human cells and in most animal cells, you can convert the DNA, or you can make a copy of the DNA into messenger RNA. So going this way, where you go from DNA into RNA, and this is critical to, and the next fact is really critical for later on in the talk to remember. But the human cells and cells in general cannot convert RNA into DNA. So you can't go back that way. So, and nature has actually, in a way, designed this to preserve the integrity of your DNA. Your DNA is your genetic material that is passed on to cells when they divide and through your offspring. So it's designed a system where you can convert DNA into RNA and then into protein, but you cannot go the other way. Okay, so that's important to bear in mind. So if you're making a recombinant vaccine, you get the, as I said, you identify the gene for the virus surface protein. You can inject that into the host cell here, and then that will make fresh messenger RNA, which is then translated. It's basically a code, and it's shown down the bottom here, the genetic code is shown at the bottom here, and that translates that code into a sequence of amino acids which make up the protein. Now, I've shown the protein here as a linear molecule, and when we write the code, it's usually displayed in a linear manner because that's how it's easy for our brains and our minds to understand all of these complex procedures. So even though we write the codes as a string of letters, in reality, inside the cell, the protein is not just a linear molecule. It will tend to fold, as shown in here, this big globule here, into very complex three-dimensional shapes. And the cells will also modify that protein by adding protein groups, by adding phosphorylation sites, methylation sites, and that's called post-translational modification. And that modification and the folding and that three-dimensional shape of the protein, later on, we'll discuss why that's so important for vaccine production. Because that shape is what is needed to give you a very good immune response and an effective vaccine. So in a recombinant vaccine, the protein is produced in a host cell, and that could be a bacteria or yeast or a human cell line in a lab, and then it's purified to make the vaccine. However, as we mentioned earlier on, some of the more traditional vaccines in the past have just relied on the fact that if you put in the virus, even alive or a killed virus, into your body, it will generate an immune response to a random part of the virus. So any part of the virus could trigger that immune response. But as we've gained knowledge over the many, many, many years of research, we've come to understand that there are key molecules on a virus which are essential for its function. And if we can target those molecules, we can make the vaccine much, much more effective than just randomly hoping we get the right vaccine in the right shape. And it's shown in this photograph down here. This is a model of SARS-CoV-19 virus. And on its surface, you see the famous so-called spike proteins. So these are a specific protein with a specific three-dimensional shape that when the virus recognizes it, so this is basically the key, what we call the key molecule, and it will only work and recognize a specific receptor on the surface of a cell for which it's designed to infect. So if you have a virus from one animal, usually it will not transfer species and infect another species because it won't recognize the receptor, or we call that's the lock and that's the key. It won't recognize the receptor on the cells and won't be able to infect that particular animal or a human being because it's a different receptor. Yes, it can change, and you can get viruses spreading from different species, but that's for another day. So in this case, you have the virus recognizing and locking in onto the surface receptor that's specific to that virus on the surface of the cell. This then lets the virus into the cell, and where inside the cell, as mentioned in the video, the virus undergoes, it will just force the cell to make hundreds and thousands of copies of itself. Now, normally viruses, they can't reproduce or make copies of themselves without a host cell. So unless they've got into another cell, they can't multiply. But once they do, as you can see, you can start making lots and lots of the virus, which then go on and infect other cells. So you can see very quickly in how easily virus could cause disease by killing thousands of cells and causing organ failure by just spreading uncontrollably through the body. So once scientists have understood more detailed of this mechanism of how the virus and the specific cell protein reacts and interacts with the lock and key model on the surface, they've been thinking, well, why don't we design antibodies that target that particular key to prevent the virus not only infecting and going into the cell, to stop it getting into the cell and also killing it. As we mentioned in the video, once the antibody sticks to the virus, it will target it for it to be destroyed. But if you target that cell key, the spike protein, you not only target it for destruction, but you stop it from entering other cells and causing the immune system, causing further infections. So this is now where scientists are looking at targeting and preventing infection rather than just having an immune response. You will still have an immune response, but you're also stopping the infection spreading. So that's the aim. As I mentioned earlier on, the three-dimensional shape of the protein is on the surface that's used, as we call the antigen, to elicit the immune response. This molecule here has to be in the right shape to generate the right antibodies that will form the exact shape to stop it sticking to the cell receptors. However, if this protein is made in host cells which are not human cells, or in bacteria or yeast, or even when it's been purified, even when it's been purified, that protein starts to degrade, and you might end up with fragments of the protein, which will still elicit an immune response, but may not elicit the right immune response to the right part of the protein. And what we need to do is to try to stabilize that protein so the vaccine is much more stable and effective. Because if you get fragments, you might have antibody reactions to all parts of the virus or none at all. And a lot of vaccines produced in the past were done by trial and error to find out which parts of that protein were causing the best immune response. And usually it's the ones that are on the surface of the protein and that form the pocket to that receptor. This brings me on nicely to have a quick discussion about the new generation of messenger RNA vaccines. So scientists have thought, well, okay, how do we get the cell, our own body and our own cells, to produce the antigen, the protein that will elicit the immune response? How can we do it better, quicker, and more safely? We can't really use DNA and inject DNA into cells. We can do it, but there's lots of ethical and moral considerations there to be injecting DNA into human cells, because once you inject it, the possibility arises that DNA could interact with the DNA that's in our own cells. So there's lots of considerations not to do that. So scientists have thought, oh, well, what other ways can we do it? And the technology wasn't there before, but over many, many years, a lot of research has gone into finding out how you can get RNA. So this is the molecule that we discussed earlier on, the messenger RNA, the one that carries just the signal from the nucleus into the cell to make a protein. How can we get the messenger RNA as a vaccine? So you don't have a virus, you don't have a protein, you have none of that. You just have a very small piece of RNA, which can then direct vaccine production. And this slide shows here where basically you can type in the code, and this is the genetic code for a gene, into a computer. it will then tell some robotics and really complex machinery to actually synthesize that RNA. And the code here will be translated into the RNA code at this end. And it's all done by machines and robots, and they can produce huge amounts of this in a very pure form, very, very, very quickly and relatively cheaply. So once we have the RNA, the next hurdle that scientists have taken a long, long, long time to overcome, but have overcome now, is how do you get that RNA into the cell so that you can then make the protein? So there are different ways, and different groups and different scientists have come up with different ways of trying to get that RNA into the cell. One way is, I've said fat droplet, but basically these are synthetic lipids that can coat the RNA and form nanoparticles. And because it's a fat droplet or what we say a lipid droplet, we know that the membrane of the cells is also lipids. So once you inject this lipid droplet into the body, the lipids will fuse with the lipids on the outside of the cell, and then the RNA can go inside the cell. The other approach used by other scientists is the use of a harmless virus, which has been modified extensively so they cannot replicate. And this is the approach used by AstraZeneca and the Oxford team to put the RNA inside a harmless virus, which cannot replicate in humans. That virus is then injected into the body, and the virus enters, just like I showed before in the previous slide, will enter the cell and release just the RNA for the protein that we want to make, the surface protein, spike protein. There's lots of advantages to this approach. As I mentioned before, and the critical phase was that you cannot go from RNA, from the messenger RNA to the DNA. So you protect the integrity of the DNA and the genetic code that's passed on from cell to cell. The other thing is RNA actually is very, very unstable. Unlike DNA, the RNA molecule is actually very, very unstable. And there was a lot of research done to try to stabilize the RNA. And as we know, we hear the vaccines produced by Moderna and Pfizer have to be stored. I think the Pfizer one has to be stored at -70. It's because the RNA in that vaccine is highly unstable and will break down quickly. But that's actually an advantage for a vaccine. You want to be able to get the RNA into the cell. The cell then produces the spike protein, and then the RNA degrades naturally very, very quickly within hours. You don't want it hanging around your body. So once the RNA is in the cell, it then produces the protein, which is then correctly modified, because this is your own cell, your own body's machinery doing the right thing, and adds the right modifications, the right kind of changes to that protein that would only happen inside the body. That doesn't usually happen in other cells or in other animals, but only human animals. You get the right folding and the right structure for the spike protein to be produced, which is it will be as close as you can get to the spike protein in the virus that you're ever going to get. And then that will then trigger the immune response, and the antibodies will bind to that spike protein, which is the key protein needed for the virus to enter the cell. And so the idea is that you not only neutralize the spike protein and any virus that has the spike protein, but also stop the virus from binding to the cell. So there's a pros and cons to messenger RNA vaccines. One of the advantages, it is so much easier to produce messenger RNA. I mean, it's really, really easy. As I said, a computer, you type in the code and the machine will produce the RNA at the end of it in hours. And we're not even talking days or weeks or months. It can do it in hours. And also that once you've put the RNA into the natural cell inside your body as a vaccine, it will produce the most natural form of that spike protein that you could ever hope for. So this will lead hopefully to greater specificity. And also, as I explained before, this is potentially safer on paper. If you look at the science behind it, it is potentially safer than any other previous vaccines. Scientists always aim to try to make it safer and more effective. It's safer because there is no virus there. There is no whole virus. There's no parts of the virus. There's not even parts of the gene of the virus that could become a virus. It's a tiny bit of just the spike protein, the protein that's needed to enter a cell. And the other really important thing, and it probably is more relevant now when we're hearing about these new virus strains or new changes to the COVID virus, this technology was designed to be able to be quickly adapted to meet any changes in the virus. So when you go back here, if the virus protein changes even a little bit here, and you need to make a new vaccine, you just type the new code in the computer here, the machines produce a new messenger RNA, you've got a new vaccine. And that can be done, again, literally in a few days, rather than having to go through years and years of research. So there are lots of advantages. Obviously, there are some, any new technology has any disadvantage. This is very new technology in terms of vaccine. And obviously, there's a lot of public concern about the technology behind it. But we know from the information from the large phase 3 studies that have been done, that generally, overall, it is safe and it is effective. There are issues about, for example, on the Pfizer vaccine, the Pfizer BioNTech vaccine, about storage and delivery, the logistics behind the vaccine delivery. The Pfizer one has to be stored very cold, minus 70 degrees, because of the lack of stability of RNA. But their vaccine contains very little anything else, and that's why it's also probably more safer. The less things you inject into somebody, the safer it's going to be. So if it's just RNA and a bit of lipid, the less likely to get you are to get some kind of negative response. The AstraZeneca Oxford University vaccine, that can be stored at 4 degrees because it's a virus, even though it's a virus that can't replicate. It's a live virus and can be stored a bit longer. The Moderna vaccine that's coming along, again, they've used a lot of science and technology to stabilize the RNA so that it can actually be stored at, I think, 4 degrees and even room temperature. So they've worked really hard on not just producing the RNA, but producing the RNA in a manner that can be stored and delivered more effective. And there are still questions that we don't know yet. We don't know how effective it is, how long the immunity lasts, and in reality, can it also stop the transmission of the virus? So there's still some unanswered questions. And I'm going to now just briefly cover some of the myths that we hear out there about the viruses, about the vaccines. And then if there are any questions and answers, I'll let people ask their own questions. So some people say, the vaccines tamper with your DNA. 75% of vaccine trial volunteers experience side effects. Beware. As I explained, and hopefully you've understood from my explanation, it is not possible for RNA to interfere with your DNA. It's just not biologically possible. Okay, you can do it in the lab, using lab conditions with a special enzyme that we use that's called reverse transcriptase. You can do it in the lab settings, but inside your body, the machinery and the necessary enzymes to do that, and all the cellular structure to convert RNA into DNA cannot happen. Does not happen. So you can't tamper with your DNA. Yes, a lot of people, volunteers, will have some side effects for any vaccine, and usually it's very mild. You might have a slight fever, irritation at the point of the immunization. Usually, it's what is in the jab is injection is some what they call carrier molecules to help stabilize the RNA. And I think in the Pfizer vaccine, it's just polyethylene glycol. And some people are allergic to that, and we know that, but it's a very small percentage of the people, as far as we know so far. Secondly, the vaccine came out too quickly. They must have cut corners on safety. Okay, so again, I hope I've tried to explain is the history of the vaccine development. This did not happen overnight. Labs across the world have been working on it, making the vaccines and the technologies behind it better, safer, and more effective. For example, the technology behind messenger RNA vaccines have been around for a while, but normally it's really the regulatory process takes 5 to 10 years, because with a vaccine, you need it to be 100% safe as possible. and it can take years to get approval, just because you're giving something to healthy people. So they need to be really, really sure that it's safe. But and there hasn't been a real need for a new vaccine really until recently, and this has driven that step forward in this technology, because we need something quickly. There are some of the vaccines coming through, and some of them are what we said, the traditional ones, the live ones like the flu jab. There are live vaccines or attenuated vaccines coming along, but as I said, they take a lot longer to get to the point where you can actually deliver it. So they're still a little bit behind the curve in terms of development. Okay, some people says we can't believe that this new claim that it's 90 to 96% effective. Well, it's hopefully, again, if I've tried to explain to you the science behind it, the fact that the messenger RNA goes into the cell and produces as natural form of that spike protein as you can get. Actually, you know, well, we were all, I think a lot of scientists were hoping and praying that we would get 70 to 80% effectiveness. That is 90 to 96 is actually a vindication of that technology. It is as good as we had always hoped, and we hope that it will carry on being as effective as it's rolled out. I mean, the effectiveness is, there's two types of effectiveness. Obviously, you want to stop further spread of the virus, so that there's a measure of effectiveness on once you've had the vaccine, can you catch it again? And there's also effectiveness in terms of, does it stop you getting a really severe form of COVID disease? And I think all the vaccines so far have shown quite a lot of effectiveness in stopping or reducing the chances of somebody getting seriously ill, even if there's a small chance some of them might catch it after they've had the vaccine. Okay, and we hear that from, we hear people saying, well, look, you know, I'm going to sit back and let others take the risk of having a vaccine. This is only going to work as a society and as a country if we all get together and take the vaccine, because there are people out there who can't have the vaccine or won't be able to have the vaccine. And as a society, like in polio and in smallpox, we need to reach a point where we're all vaccinated to the point where the virus can protect those who can't have the vaccine. So, you know, I think that's a very selfish point of view, personally. We're here to try to, you know, when you take the vaccine, you know, you're not only helping yourself, you're helping others as well. There's also been claims that the vaccine contains cells of aborted fetuses. Not true at all. As I explained, the technology explained to you before is the new ones, there are synthetic RNA. It's a chemical reaction that makes the RNA. They don't go anywhere near any cells. They don't go near any virus. They don't go near any aborted fetuses. And so none of the vaccines, as far as we can tell, contain any cells or tissues from human or animal cells. There's also a claim that vaccines contain pork animal products and gelatin. That may have been the case with some of the older vaccines, and sometimes, you know, they were there traditionally from a long time ago, and they've not changed, because usually if you change even a small part of that vaccine, you know, remove the gelatin, for example, you have to go through a whole 5 to 10 year cycle of approval before that vaccine can be released. And a lot of companies and pharmaceutical companies can be resistant to that change because it is a long, lengthy, expensive process. But with this change, with COVID, we have had to be forced to make new technology available. And as far as we know, the scientists have taken into account all of this, and the vaccines don't contain any animal products or byproducts. So this just leaves me, I'm sorry this took longer than I thought, but just leaves me to open up to questions and answers. So I'm going to unstop sharing, and hopefully we'll be able to hear some questions and answers.
So thank you very much, Raj, for that. Now I'm going to stop the recording now, and